So thank you everyone for being here today. My name is Pia Rodriguez and I'm here with Dr. Shi Wang and Luke Bartlett. We are all graduate students and a postdoc fellow from Duke University, and we're very excited to welcome you to our first week of our Duke Chan Zuckerberg Initiative Bench to Bedside virtual speaker series. So to give you a little bit of background of why we're here today, we are sponsored by the Chan Zuckerberg Initiative Community Building Grant. And with their support, we identified the opportunity to build a network among academic researchers, biotech researchers, patients, and clinicians working in the field of neurodegeneration right here in Research Triangle Park, North Carolina. We really believe, as I'm sure many of you do, that the key to combating neurodegenerative disease lies at the junction and collaboration between these groups. So in order to kick off building this community, we brought together 4 expert representatives from each of these groups to share their experiences, challenges, and ideas for collaboration in the neurodegeneration field. We hope this speaker series will bring together our community and connect us for future collaborations. So before I introduce our first keynote speaker, Dr. John Didsbury, I wanted to go over some brief logistics with you. After today's talk, we will have an open Q&A session followed by Zoom breakout groups to better network and discuss ideas. We ask that you please try to keep your video on during the Q&A and breakout groups in order to foster, a more personal sense of community, especially now that everything is virtual. If you prefer, please feel free to drop in questions using the Zoom chat. Or use our Slack channel for discussion. If any of you have not yet joined the Slack channel, I will drop the link to our Slack channel in the chat shortly. And we are really thankful for your participation today. We know that you all are busy and coming from very many different backgrounds, but we're excited as trainees to have had this opportunity to get started in building this community. So we really thank you for your support. So with that in mind, I wanted to introduce Dr. Didsbury, who is our first speaker for this series of the month. Dr. John Didsbury is the president and CEO of T3D Therapeutics, a pharmaceutical company right here in North Carolina's Research Triangle Park, using a transformational approach to treating Alzheimer's disease. T3D is currently in a phase 2 pioneer study, which stands for Prospective Therapy to Inhibit and Overcome Alzheimer's Disease Neurodegeneration via Brain Energetic and Metabolism Restoration. Dr. Didsbury is a seasoned executive manager with over 28 years of experience within the pharmaceutical and biotechnology industries in both small and large public and private companies. Dr. Didsbury was president and CSO of DARA BioSciences Incorporated, where he led the development of T3D 959 and directed all drug discovery programs of the company in his chief scientific officer role. He played a key role in taking the company public in 2008 through a reverse merger. Previously, Dr. Didsbury served as Associate Director of Biology at Macronex Incorporated, as Assistant Professor of Medicine at Duke University Medical Center, and as a scientist at Genentech Incorporated. So with all of this wonderful years of experience, we're very excited to have Dr. Didsbury join us today and share his perspectives from biotech in the neurodegenerative field. Thank you so much, and we hand it off to you, Dr. Didsbury.
Yeah, thank you for that kind introduction, and welcome everyone. It's a real pleasure to be talking to the Duke community once again. It's near and dear to my heart, having worked there for a number of years back when we had a basketball team that won back-to-back national championships. So let me move to the next slide, and this is what I'm going to talk about today. I want to introduce you first to our company, where we are treating neurodegenerative diseases from a metabolic perspective, which is unlike many others in the field. And then I've been asked to speak largely to have this talk focus on the benefits of collaboration. And so I'll give you my perspectives from the pharma biotech industry on what those benefits are for each of the key potential collaborators, partners in any collaboration. And then I'll talk about specifically what has worked and what hasn't worked for our company in terms of collaborations with various groups. And then to kick off a real Q&A towards the latter part of the talk, I'm going to give you three major challenges to success for our company. And then what clinicians, patients, academic researchers, and other industry researchers can offer to help meet those three key challenges. So in the next slide, this is a bit of background and rationale for why we believe that Alzheimer's disease is actually a metabolic disease. A lot of people are looking for new therapies to try to treat this disease and approaching drug development as trying to find the top of a pyramid. And that if you do so, you will evoke changes downstream from the top of the pyramid in a cascading kind of manner. I'd like to have people change their mindset and look at it differently. And the basis for this is that there are three major hypotheses for the disease. One, which is what we adhere to, is that the key part are metabolism changes, particularly glucose metabolism and lipid metabolism dysfunctions in Alzheimer's disease. Secondly, there are, as everyone is most aware of, structural changes, amyloid plaques and tau tangles, to name a few. And then there are also stress changes, inflammation and reactive oxygen intermediates, oxidative stress. There is evidence supporting all three of these hypotheses, but our hypothesis, the neurometabolic hypothesis, is that the metabolism changes may be the initiating event. So if we instead, in this next slide, I want you to look at this disease not as a pyramid of events, but as a massive interconnected positive feedback loop where each of these changes is interconnected with the other, and these changes actually play into each other. And so whereas most people were going after structural changes and failing, very few companies are going after glucose and lipid metabolism. There's a growing interest, as you may well be aware of, in stress changes, particularly inflammation. And I'll give you my perspectives on that. Hold on a second, I just want to make sure, yeah. In looking at this, one of the things that people, just a few key points here, stress changes and inflammation. So when the beta amyloid plaque hypothesis is really falling, in my mind, falling out of favor as a key upstream event, and most recently with the failure of AC Immune Roche's anti-tau therapy, this clearly may not be the upstream initiating event to be going after. And so the field is moving towards inflammation. And part of it is that inflammation is easy to study. And it's kind of a herd mentality that I see, and people moving towards looking at this because there are a lot of targets that one can test, and it's more easily studied. Metabolism has, is very different. And the reason we think metabolism is important is that you take a step backwards and look at the brain. The brain is the highest energy expending organ by tenfold on a weight per weight basis than any other organ in the body, where glucose is nearly the sole energy source for this highly metabolic organ. And secondly is that the brain is composed of half lipid. And so the lipids are very key for structure and function of the human brain. And these two observations actually point to a potential vulnerability points to dysfunction and disease because of this high metabolism and the high lipid content in the brain. So this is what we are looking to do with 959 is to correct glucose and lipid metabolism and break this feedback loop at this particular intervention point. So our drug, 959, is a small molecule. It's an agonist of two targets, PPAR delta, and the secondary target is PPAR gamma. Both of these targets regulate glucose and lipid metabolism, and they function. These are nuclear receptors that function to maintain homeostasis. The molecule is brain penetrating, which is very important since only 2% of all drugs for all diseases are able to penetrate the brain and cross the blood-brain barrier. The other beautiful aspect of this drug is that it's given orally, no injections, no IV infusions, and it's once a day administration. A key aspect here is that this is uniquely different than another class of molecules which you may have heard about for treating type 2 diabetes, the thiazolidinediones, which are PPAR gamma selective. And PPAR gamma has restricted expression in the brain. So examples of those diabetic drugs are Actos and Avandia. The primary target, PPAR delta, is highly and ubiquitously expressed in the brain, and it's particularly found highest in areas of the highest energy metabolism, and it acts to overcome brain insulin resistance to restore normal metabolism. And so this is a repositioned new chemical entity, repositioning this from being pursued for type 2 diabetes to now Alzheimer's disease. So this just kind of gives you a schematic of what's going on here. Delta regulates lipid metabolism and glucose metabolism, whereas PPAR gamma regulates more glucose metabolism. And to put it really simply, PPAR delta regulates energy expenditure and PPAR gamma regulates energy storage. Lipid and glucose metabolism are intimately associated with the formation of plaques, tangles, inflammation, cell loss, myelin loss, vascular and synapse damage. So these are very key aberrant metabolism in terms of lipid and glucose can really play a role in virtually every pathological sequelae in Alzheimer's disease. A little bit of the history of this compound. It was being developed in the late 2000s for the treatment of type 2 diabetes. The company got it to a certain point, and I had left the company at that point, and was developing it for diabetes with two key attributes, differentiators from Avandia and Actos. One, it had no cardiotox, and two, it actually could improve diabetic dyslipidemia. We had preclinical data on this molecule showing great efficacy in multiple diabetes and dyslipidemia models, and it has shown excellent efficacy in an intracerebral streptozotocin model of sporadic Alzheimer's disease. We established rat brain penetration, and we have finished our long-term rat and monkey GLP tox studies, which now allows us to dose a human being for any length of time. And we have a very large therapeutic window. So we are dosing humans well below the no observed adverse event level in those long-term animal safety studies. Phase 1 was a stellar phase 1, excellent PK, good exposures. We didn't even reach a maximum tolerated dose, so had an excellent safety profile. We ran a phase 2A mild to moderate Alzheimer's subjects. This was just a 2-week dose range finding study. We didn't have a placebo, but it did give us a lot of useful information on a shoestring budget. Excellent safety profile. There were no severe adverse events. We had multiple efficacy signals. We saw changes in glucose metabolism in the brain as measured via FDG PET. We saw improvements in cognition as measured by ADAS-Cog11. And executive function as measured by DSST test, and changes in plasma metabolomics that indicated that we were regulating in a positive manner both glucose and lipid metabolism. Now, this has led us to do a major phase 2 study that Pia told you the acronym for, the PIONEER study. It's a randomized, double-blind, placebo-controlled study, and we're using 40 to 50 US sites. We are looking at safety, efficacy, and about 25 different blood-based biomarkers. We're not doing CSF biomarkers to minimize patient burden and avoid having not subjecting them to multiple lumbar punctures. And the subjects have mild to moderate AD, and we'll be treating with 3 different doses of drug versus placebo, and having roughly equivalent ApoE4 positive and negative genotypes in each arm. And we'll be dosing these subjects once daily for 24 weeks. Now, we had just a little bit of information. We started screening our first patient in the first week in February. And we were projecting to complete enrolling of 256 subjects by the end of the year, in roughly 9 months. And we were actually going at such a fast rate, enrollment here, there was a very high interest in getting into this study that we were now projecting full enrollment in 5 months, and then the pandemic hit. And so this compromised operationally a number of clinical sites such that we could not ensure the safety of patients. Either the patients, namely the patients couldn't get in the clinic to be seen. And so we had 33 subjects on drug at that point, and so we had to put a temporary halt to the study. We are now adapting this protocol to so that in the event of any major surge in this pandemic, we'll be able to complete the study. And that's a subject for another talk, but basically we are creating a remote setting within the clinic, so using 2 separate rooms, and such that if the patient can no longer make it in or the clinic is not open, that second room at the clinic now becomes the home, and the patient is already used to being administered tests via Zoom type interface. So let me move to the, I think I'm okay on time, Pia. I haven't checked. Benefits of the collaboration of the biotech companies. So this is my opinion, not to be construed as fact. But collaboration with clinicians, collaborating with clinicians is really beneficial in terms of it provides a bridge between the customer and the developer, the patients and the biotech company. Through clinicians, we can identify the unmet medical needs of patients and therefore tailor or work on therapies that'll meet those needs and that those are realistic needs to be addressed. It also allows us, as you're going through finishing up small trials like we did in the phase 2A, it gave us the opportunity to just kind of ask them, well, how did this therapy do? And the feedback you get, while it's anecdotal, is important, and it is not captured in these scales to measure, test performance. But it's important to kind of determine where you want to go. And what we found, for example, in talking with clinicians was, one of the things that we observed was the drug maintained its effect. Peak effectiveness was actually occurred a week and a half after dosing was stopped. And then we found out, through this type of clinician feedback that the effect lasted about 6 weeks after therapy was stopped. The patient was back to where they were before dosing started. So it gives you a handle on how often you might have to dose your subjects and at what level in future studies. In terms of patients, they're an inspiration to the biotech company. After the 2A study, I had caregivers insisting on meeting with me and hugging and crying and saying that this drug, even though it was only 2 weeks dosing, literally changed my loved one. And boy, if that doesn't pull at the heartstrings and get you to stay the course, nothing does. Patients also can be a good group voice to enact change, can influence granting agencies that will provide new capital for companies, and also to put pressure on payer reimbursement policies to improve the economics of drug development for biotech companies. In other ways, it can enhance clinical trial enrollment, spreading the word. That clearly happened at one site that we had in Charlotte. Word got around rapidly, and people were coming in left and right wanting to try to get into the trial. And then patients can be very valuable for enhancing value in post-approval marketing efforts, as you see patients being interviewed on various commercials. With academic researchers, the benefits of collaboration really is supportive research for the efforts of the biotech company at a relatively low extra costs, provides the company potential to generate new patents, enhance company awareness, and academic researchers who are key opinion leaders, aka KOLs, really enhance company optics and provide validation of the company to investors for future capital raises. Other biotech benefits of collaborating with other researchers at other biotech companies include really generating mutually beneficial data, if they can get a collaborative program together where each gets information that is helpful to the other. It is also the opportunity to identify if the potential for combination products at each company that may be of value. And it may be so good that it provides the foundation for a future merger acquisition between the companies. In terms of our company, what has worked, not worked, and why? Well, let me start with the clinicians and just a few points. What has worked are things like, and one prime example is a new added value direction for the company was identified. And that is Huntington's disease, where the mutant Huntingtin protein actually interacts with the primary target of our drug, PPAR delta. And so that is an area that we are now pursuing as a second indication for our lead molecule. What has not worked, and this is, not especially, attributed just to clinicians, is timeliness. They have their day job too, and clinicians are oftentimes very busy. And so trying to fit in collaborative work can be a challenge. In terms of patients, what has worked, as I pointed out, inspiration actually provided leads to new investors. These people know people and can actually point the direction of others who may want to support your endeavor. And as I pointed out, can provide insights into aspects of drug activity. And what was nice was that we received, after the 2A trial, a number of unsolicited caregiver patient feedbacks that give us a lot of optimism in moving forward and to actually make sure we pick the right kind of scales to look at specific aspects of cognition and function and apathy and so on in subsequent trials. I think what has not worked with patient is that there are different expectations. Getting anyone to understand the complexities of pharmaceutical development and that things just cannot happen quickly when you've got 10 different functions, that has not worked with patients. In terms of academic researchers, I think what has worked is non-clinical data generated that supports the thesis behind projects at the biotech companies. What has not worked is oftentimes, academic researchers have a different agenda than biotech researchers. And so oftentimes the academic researchers have different priorities. There is a mindset that I observed of research for research sake, which is clearly not aligned with research for bringing new drugs to market. And the ideal experiments for drug developers oftentimes are not carried out in these sponsored research agreements, where to the drug developer to get dose response relationships established is very important. To the academic researcher, it may be just timing. They want to look at this aspect, when you started giving the drug and so on and so forth. So there's different priorities. And what has not worked with academic researchers is university tech transfer offices that, I got a typo in there. There is overreaching and overvaluing the collaborative output. Wanting, license deals that, it's way too early to even be asking for a piece of the pie. and for molecules that are already well into the clinic to just get a little bit of supportive data here and there, you know, it's not worth as much as the tech transfer offices believe. Working with other biotech researchers, this really is not applicable for our company, but I think what can work, I'll put it this way, are shared learnings between these researchers for improving therapeutic development. It provides a greater awareness of the competitive landscape to know how you are going to have to differentiate your product from a competitor's. And what may not work is you have to be careful is loss of intellectual property protection. If you are giving your therapeutic to be tested by the other biotech researcher, and there is always the potential of that other company reverse engineering and finding out the structure of what they have been given. So you have to just be careful on that front.
Hi, John.
Yes.
Sorry, I wanted to let you know that we have about 1 to 2 minutes left, so we can move on to questions.
Okay.
And we have question slides, so
okay.
yeah, thank you.
Okay, thanks for that heads up. Okay, so I'll talk a little quicker. I see 3 major challenges to success in the neurodegeneration field and others, okay? It's not specific to neurodegeneration. One is siloed science. There's a lot of specialization within each scientific field that can lead to tunnel vision. And these knowledge firewalls limit cross-fertilization of ideas, particularly in our case. No one even really understands metabolism. The neurologists don't, and the metabolic folks don't understand neurology. And I've experienced kind of a country club attitude in that if you're bringing in new thinking, you're treated as a non-member and it's hard to get into that club. The second is what I call superficial science. I see poor reader due diligence, skimming rather than really understanding whether an article is real. The data is really of value and reproducible. And that's compounded by news media simplifications and you can read it. The other is a feeling of a major challenge is risk aversion in neuroscience. It's a relatively nascent field. The brain is one of the last to be explored frontiers in terms of organ systems in the human body. And current therapeutics effectiveness is marginal in the field. And so this just leads into the discussion slide where I haven't really filled anything in what the collaborations can do to address these major challenges. And so with that, I will end the show and stop sharing.
Wonderful. Thank you so much. That was great. If everyone could please give Dr. Didsbury a round of applause virtually as well. And so you've given us a lot to think about. I think that was a great starting point for this community to think about the barriers and where we can really come together. So if I could please ask anyone that is willing to turn on their video during this discussion, we will have open questions for about 10 minutes and then go off into smaller breakout rooms. So Dr. Didsbury, we have a question,
okay.
2 questions actually, and I'll read them. They're from the chat. So you talked about T3D959 as a potential drug for treating mild to moderate AD. One question is, can you envision the drug being used to prevent AD in the future?
Excellent question. It is very, to prove prevention is not really, I believe it could prevent. It really is altering the way you use energy to benefit the brain. To do prevention trials, though, for a small company like ours, this is going to be a major pharmaceutical post-marketing endeavor. It'll take thousands of patients in 5 years, a 5-year trial to demonstrate.
Okay, thank you. And another follow-up question to that was, because metabolic implications are shared across neurodegenerative diseases, do you think that this drug could potentially be used to treat, say, Parkinson's disease in the future?
Another great question, and it's a resounding yes. There is aberrant glucose metabolism in so many neurodegenerative diseases. PD, HD, and some of the rare orphan diseases. So yes, I do believe this drug could have effectiveness in a number of these other diseases. Here's the perspective from the small biotech company, though, is you don't want to be all things to all people, and you become unbelievable. Okay, if you say, "Oh, I can treat everything," and so you do have to focus. And so we focused on Alzheimer's disease largely because of a paper that came out in 2012. It was one that cemented my idea that this would be 2 + 2 = 20 for Alzheimer's. And that was the PPAR delta null mouse, whose phenotype had many attributes of Alzheimer's disease, and people weren't recognizing this. And so a lot of impetus, and it's just like, when you're an outsider looking in, I'm a metabolic guy. I don't know anything about neurology, but I'm looking at the brain from a real simple perspective. Okay. This is a fine-tuned Lamborghini that if it goes out of tune, it's using up so much energy, glucose-wise, it's going to throw everything into dysfunction. And the same with the metabolism, the lipid metabolism aspect. So we had to focus. And the other avenue, we picked a backup avenue, so to speak, in Huntington's disease. And that may actually have a stronger scientific rationale target-wise than Alzheimer's disease, but we will see.
Great, thank you. We do have a couple more questions in the chat. If participants are willing, you are welcome to unmute yourself and ask your own questions. We have a question from Lori and Michelle or Michael. If you want, I can ask it for you, or you can ask it yourself.
Sure, you did. So I was going to ask if you could just comment on PPAR delta expression or activation inhibition and its relevance for cancer growth in vivo and the controversies surrounding that area.
Yes. We have not seen in any of, so is this, the biggest question, is this a target mechanism, okay, or is it really compound related? And it could very well be compound related. Whereas a straight delta agonist was stopped because of potential carcinogenicity in mice, okay, that has not been seen with this drug. And it is a unique selectivity profile that there's something that people don't realize, and it's never been published. When I was at GlaxoSmithKline and head of metabolic disease strategy, we had an active PPAR program, and we knew from animal studies that if you had a molecule that activated both delta and gamma, delta counteracted gamma side effects and gamma counteracted delta side effects to give you a very unique, clean molecule. Now, the biggest risk is probably colorectal cancer, okay, because of the highest expression of delta there. And this drug has been tested in numerous colorectal cancer cell lines, no proliferation, no activity in any xenograft models accordingly.
Thank you.
Great, and we'll take one last question. It's in the chat from Michelle. Michelle, would you like to ask your question yourself, or we can read it for you? All right, I will ask it on their behalf. What is the blood sugar profile of people with AD? Will your drug potentially interfere with glucose metabolism in non-diabetic AD patients?
Excellent question. Answer, no. It really kicks in when there is aberrant glucose metabolism. This is, activating either of these targets actually does not cause hypoglycemia. There's no added risk combining it with other anti-diabetic medications. In the phase 2A, in the mild to moderate AD subjects, nobody was diabetic, but they were had fairly high blood sugar levels. I think in 90, I think in mgs per deciliter, right? In the 90 to 100 range. We did see a very nice small dose-dependent decrease in fasting plasma glucose, but nothing that made anything subnormal. It actually worked to restore normalcy for these subjects. Excellent question.
Yeah, that was a great question. So we're gonna thank you for answering those questions, and we're gonna move on to the breakout group component. So for this breakout group, I will quickly share my screen. We have a few questions for us to think about. So if everyone can see my screen, hopefully. Can everyone see my screen?
Yep.
Okay, great. Dr. Didsbury has given us a lot to think about. I think that your analysis and outlook on what has worked and what hasn't worked, I think is a great place to get started. And so we're going to break out into groups and address these three questions. Dr. Didsbury, she and Luke will be entering groups to guide discussions, but if any of them are not in your group, please proceed and chat. Our hope is that these groups will be a healthy mix between representatives of the patient community, the clinician community, academic and biotech researchers, and that you can provide each other with diverse feedback in answering these questions, because they are very complex, I think, big problems to solve. So with that, Luke will break us up into groups. And we will drop these questions in the chat, but please also try to make note of them. Okay, here we are. Yes, please try to make note of them as we go off into groups.